Hello and good afternoon and good morning, everyone. I'm Dave Iverson, the contributing editor at the Michael J. Fox Foundation and the moderator for today's Third Thursday webinar. As many of you know who have joined us for these webinars in the past, we do them the third Thursday of every month, 12 Eastern, 11 Central, 10 Mountain, and 9 Pacific. And each month we take up a topic of importance to the Parkinson's community. So we hope you'll continue to mark your calendars and join us throughout the year. You can join us both live, and you can also watch these on demand later on. If you would like to submit a question throughout the hour, which we will answer, and starting in about 40 minutes or so, you can just use the box, the Q&A box that you see near the middle of your screen. You can type in your questions there, and we'll do our best, as always, to get to as many of those as we can. And then if we don't get to your question, or you want to still find out more at the conclusion of today's webinar, consider visiting the Michael J. Fox Foundation Facebook page on Monday at 12 noon Eastern, where we'll do a follow-up live Q&A on this topic as well. And one other note before we get started, we're also providing the slides from today's webinar for download. So you'll see a box called Resource List on your screen, and you can click on the link there, and the document will open in a new browser window, and you can save or print the slides from there. Okay, let's get started on today's topic, which is on stem cell research. Here's some of what we're going to be trying to cover on today's seminar. Actually, I'm going to ask my colleague Maggie to go ahead and go to the next slide, because mine doesn't seem to be functioning. And here we go. Here's what we'll be covering on today's seminar. We're going to talk really about the subject of stem cell science, something that's been in the news off and on for the last 15 years or so, and we really want to take this opportunity to dig into this topic and explain exactly what stem cells are, where they come from, how they work, and of course, importantly for the subject of today's webinar, how they might work to treat people with Parkinson's disease, and how they're also being used, importantly, to give us a deeper understanding of the disease itself. Let's go ahead and meet who's going to be participating in our subject today. Joining us is Dr. Ulla Isaacson. Dr. Isaacson is a professor of neurology and neuroscience at the Harvard Medical School, also director of the Neurogeneration Research Institute there, and a faculty member of the Harvard Stem Cell Institute. Dr. Isaacson, thanks so much for joining us on today's webinar.
Thank you for inviting me.
It's a pleasure to have you. And joining us as well is Dr. Todd Scherer. Todd is the CEO of the Michael J. Fox Foundation. Todd, thanks for being part of our conversation, too.
Thanks, Dave, and thank you, Dr. Isaacson, for also participating, and I look forward to the webinar.
Thank you. And as many of you know who've participated in this past, we typically have a patient panelist as well. But because of the research and scientific focus of today's webinar, and also because, as you'll learn later in the webinar, human clinical trials of the science we're going to be describing have not yet been approved by the FDA, there isn't a patient representative in today's panel. So let's get started with our topic and with what we want to begin with first, which is really a definition of what stem cells actually are. And you see a diagram on the screen which basically says that stem cells can do two different things: replicating themselves, or they can actually become any cell in the body. So Todd Scherer, why don't you start us out with the first part of that definition, which is the kind of replenishing quality that stem cells have. This means, as I understand it, that like an adult, a blood stem cell can replicate itself, or a liver cell can replicate itself. Is that correct?
That's right. I think when we think about stem cells, it's good to remember that these are natural cells that we all have in our body from the time that we're conceived all the way through our entire lifetime. And many of our organs throughout our lifespan are constantly being replenished and rejuvenating, and that is the role that stem cells play throughout our body.
And then the other key thing, and we'll be really digging into this throughout our topic, Dr. Isaacson, is that early on, stem cells are cells that can differentiate and become really any cell in the body.
That is correct. The stem cell is just like Todd said, it can make very specific cells throughout your lifetime. But at the beginning, when the fertilized egg is present, the cells are omnipotent. They can make any of those progenitor cells for organs. And in fact, at some point, that capacity is lost. And all the stem cells, we call adult stem cells, then have very specific opportunities or tasks, such as replacing the skin every 12 days or so, or the bone over a 20-year period, or and so on. But the brain doesn't do that, except in the hippocampus.
Let's dig into that a little bit in a little bit more detail, which is what our next slide will allow us to do. And here we'll see the two different kinds of cells that the two of you are beginning to talk about. And we'll talk first about the embryonic stem cells, and then in a moment a little bit more about adult stem cells. Dr. Isaacson, stay with this then, if you would, and describe how we in research use these embryonic stem cells that come initially from fertilized eggs that are no longer going to be used in an in vitro clinic normally, and then are donated for research. Describe what those cells are and what they can become.
Well, it is fairly easy to imagine that the fertilized egg will become every cell that we know in the body, but the discovery was that the fertilized egg, to some extent, could be grown to have those cells separated. So after a few days in a dish, one can actually isolate the embryonic stem cell. And the embryonic stem cell has basically not determined yet what it's going to be. And so what we look at genetically is that most of the DNA doesn't have any instructions or marks, as we call them. So as the slide shows, because of the advent of in vitro fertilization, there are many eggs like that that are stored, and they can be donated for research or otherwise.
And as we'll hear a little bit.
With the adult?
I'm sorry, go ahead.
Go ahead.
I was just going to ask about.
Yeah.
As we will explore throughout our webinar today, these are the cells that from the late 1990s until 2007 or 8 or so were the primary focus for research, and these were the ones that researchers like you and others were then trying to develop into the kinds of dopamine cells that would be useful potentially in Parkinson's research.
Right. So
Dave, I was just going to say, as a sort of an analogy the way I've kind of thought of the distinction between an embryonic stem cell and an adult stem cell, as Dr. Isaacson was saying, the embryonic stem cell has full potential to become any cell in the body, and the adult stem cell is more limited to perhaps only becoming a certain type of cell, like a liver cell or a blood cell. The embryonic cell is sort of like the day you showed up in your freshman year at college and you were full of potential and you could have chosen any major and gone down any career path and you hadn't failed a class yet and hadn't made any mistakes in your life that you were about to make. And then along the way, you sort of lose that potential as you go down every turn and you select a major. Your transcript becomes what it becomes, and by the end, this is just being a little bit facetious, you become more like an adult stem cell with your philosophy degree, and maybe you're not going to become that doctor, so you have more limited potential. But I just think to kind of put it more in context, the really great potential of that embryonic stem cell is that potential to really be utilized using exquisite technology to direct that cell to be what any type of cell that the body has ultimately, because we all do start in that one cell form, and then that sort of evolves and branches off into every type of function that the body has.
Yeah.
No, it's a useful and nice analogy, Todd. Thank you. And if you could just take it a beat further, if you would, Dr. Isaacson, and you mentioned this on the last slide, but I think this is really a key point. If we look down on the adult stem cell part of this slide, and we see that third point, that brain stem cells are only found in one area of the brain. So unlike the liver, for example, where you can lose a lot of your liver and the liver can replenish itself through liver stem cells, the brain can't do that, except in that one area you mentioned called the hippocampus. And that's why this is so critical, because in Parkinson's disease, the dopamine cells go missing and they can't replenish themselves. So that's why we're so interested in embryonic and in another form of stem cells we'll get to in a moment, because those would be the ones that we might be able to use in a potential therapy. So pick up on that thought a little bit more, if you would, Dr.
I mean, to pick up on also on the helpful analogy by Todd, it's more actually like when a child enters daycare. I mean, these are the embryonic stem cells have absolutely no instruction at the time. The big confusion that actually from history gives us a good lesson was that in the 90s and the 2000s, there was actually debate, should we use adult stem cells or embryonic stem cells? And thankfully, science solved that issue very clearly. When we used adult stem cells, we could never, ever make them into the specific dopamine neuron, and the very specific dopamine neuron that dies in Parkinson's disease and actually makes the function of initiating movements, unless we took away those memory marks on the DNA. So the field was sort of in a flux for about 10 years, and now it's settled down, and people, as you say, have realized that of all the species on the planet, maybe monkeys and humans have the smallest number of adult stem cells in the brain, and as you say, in the hippocampus, which is a region that's very important for imprinting memory every day. It seems like evolution has played this in a way that you have to have new cells every 30 days. But the adult brain seems to work entirely on its electrical activity and its connectivity. You may know the word synapse, which means the connections between cells. So that was almost a decade of very active research, and now we've come to the understanding that the adult stem cells have very specific and limited functions. And perhaps you may have heard that we first thought that there were stem cells in the nose. In fact, you have dopamine cells in the nose, and they were studied in exquisite detail in rodents. It turns out that in humans and primates, there's very little of those new cells formed, and so when we lose our smell, it doesn't regenerate. So this field has been very active for a couple of decades, as you hinted at, Dave, and some of the things that we thought were just going to be easy, people said, why don't you just use adult stem cells and not embryonic? Turned out it wasn't that easy, and the very, very detailed research that several scientific groups did proved that you basically had to go back to the embryonic stem cell stage if you really wanted the specific cell you wanted, not just any tissue progenitor.
Well, let's move to our next slide, if we might, which takes us just a little bit further into the current research that's going on. And you see up on the screen now this idea of induced pluripotent stem cells. By way of background for this, Todd Scher, before we go through what exactly pluripotent stem cells are, one of the reasons why this advance was exciting is that as promising as those embryonic stem cells were, in the early research, laboratory research that was done in the first part of the 2000s, there were some bumps that took place. One of them being that because they come from an egg that is not yours, there can be immunosuppression problems, rejection problems, once that's transplanted into a laboratory animal, for example. So there were some obstacles, right? And that's part of what makes this new version, which we'll now discuss, so promising, because it comes from the patient themselves.
Yeah, so a couple of points in response to those comments. First, and I think we're going to get into this a little bit more in the discussion today, that the goal of much of stem cell therapy, and particularly as it relates to Parkinson's, would involve a transplant and replacement of the cells lost in the disease. So to utilize stem cells to recreate the cells that are lost in the disease and then transplant them back into the brain to rejuvenate and correct the problem that occurred because of the disease. So just like with any type of transplant, there's always a concern, which is at this point, I would say, still a hypothesis that there would be a concern in terms of the matching of that transplant, in terms of whether there would be immune rejection or any immune system issues in terms of how that transplant would be taken up by the patient. And like I said, that is at this point, I would say, still to be proven. It's not clear that that would actually be the case. I think the other thing where there's
let me clarify that one. I mean, as Dave said, if you take a non-matched tissue outside of the brain, at least, and you place it in another person's body, it will reject within 10 minutes usually, unless you have very strong immune suppression. You may be able to, and all the so-called non-self transplants that are out there so far, even in the brain tissue, they always show an immune reaction that we can measure and we can test, and we can see antibody formation in the blood of every patient or every animal we do. But as you point out, if you provide immune suppression, you may be able to overcome that. But the fact of the matter is that self-autologous does, as far as all tests today show, will not create an antibody reaction. But if you use another type of tissue from non-self or from another patient, you will have some form of immune reaction. The question is whether it will block function or not.
So with these new induced pluripotent stem cells, which was this breakthrough that took place in 2007 or 8, the idea for these was revolutionary. Walk us through, Dr. Isaacson, if you would. This chart, I think, does a pretty nice job of explaining how it happens. But just take us through that process, and then, Todd, perhaps you can pick up on what some of the real advantages, potentially, what some of the real advantages of this process are over the embryonic stem cells we mentioned earlier. But Dr. Isaacson, sort of walk us through a bit of the diagram.
So the history is quite extraordinary. Shinya Yamanaka in Kyoto, Japan, was working actually originally in many fields, but a little bit in cancer and a few other surgical fields, when he basically was looking for a code to see how young a cell was. And he was also adding factors to see if he could, induce them to become less determined, as we say, less defined. He came across 3 factors that stripped the DNA of its coding, so it became sort of a naked form of its original self. And what he basically revolutionized, and not surprising, he got the Nobel Prize at a very early age and almost a few years after his discovery, was that we had all believed that once a cell is determined, it's fixed. But he managed to show that if you push the cell back with those factors, it will actually determine itself that it is a stem cell, like we just heard. It will keep dividing without these factors that Shinya Yamanaka provided them. And as you can see on the diagram, they're now reprogrammed. They have, for all intents and purposes, even if we do a big gene expression profile, we cannot determine, say, between 10 embryonic stem cells and 10 iPS cells, any really significant categorical differences. And so it was revolutionary to all of us, and some of us were fortunate to immediately be able to use the technology and then make these dopamine cells that we had spent the few years before using from the embryonic stems. So it can give any individual an opportunity from almost now any cell, blood, skin, or any cell that is in your body, when treated with these factors for a week or so, the cells will, one of many cells will revert back to this state, and then you can pick them up.
And touch here, as I understand it, then one of the real great possibilities here, and again, we're not yet to the point where these are actually being used in clinical trials with human subjects, but the idea is so enticing in part because not only can these cells become any cell in the body, they're your cells. They're potentially something that represents a kind of personalized approach to this and would get around the possible problem of an immune rejection. Is that right?
Yeah, so there's really 2 exciting aspects of this. One, which is what you just mentioned, which is that you actually would be theoretically then able to do the therapy and the transplant with the individual's own cells. So you would really overcome some of those challenges that we just discussed. And I think secondly, what's really exciting about this is the ability to then as well get a better insight into the disease by studying these cells that are coming from people with Parkinson's, that we can, in fact, take the cells and make them into brain cells and then try to understand what might be happening in the context of Parkinson's. So, I think the other point I wanted to make related to this is that it's really a great example, the discovery of iPS cells, about following the path of scientific investigation, and where we started with our work in embryonic stem cells, which is still advancing. And this new discovery sort of was based on a lot of the really fundamental and elegant science that was conducted through the early work in stem cells. And just to kind of reiterate the important need of continuing to support and have financial resources for this fundamental research without any sort of restrictions, because you never know where it will lead. And here we have such a great outcome.
Yeah, I'm glad you underlined that point, because we can't get to where we want to go, the eventual applied goal, if we don't engage in this kind of important fundamental basic research. Let's get to the heart of how these, we hope, might someday be used in Parkinson's disease. So let's go ahead and take a look at our next slide here, where we actually we went one too far, so we'll go back. And now we're one back too far, so we'll go ahead once more. And here's where we want to be. How we can actually use these to treat Parkinson's disease. So we've got these cells that can become any cell in the body. And importantly, the next step, Dr. Isaacson, is to develop these into dopamine-producing cells, because we're not going to transplant stem cells into the body. We're going to transplant the cell that makes the dopamine that is needed in Parkinson's disease. That's at least the goal. So the first step really is to coax them into being the right kind of cell, right?
Yeah, so like Torchero just said, this really came from very, very basic research done in the 80s, and I was fortunate to participate in that too. But the short of the long story is that the dopamine neurons that we developed early on to replace the very specific ones, there are 5 or 6 different dopamine cells in your brain, by the way, so it's not really, a transmitter disease. It's a disease of a very specific nerve cell and its connections. So over the next few decades, we started with fetal cells in very simple mice rat models, and we were able to show very convincingly that these implanted cells grew up, and when they were able to release dopamine, as is the second point there, synaptically, we would always see the behavioral response. We never saw it one day after transplantation, but also always depending on the actual species used. So if you transplanted rat cells, it was 21 days. If you transplanted, say, a pig, it was a little bit later, monkeys 6 months, and human dopamine cells took 9 months to basically grow up. So there was massive evidence that we could actually control and predict what the outcome was. However, as with all new therapies, some of these therapies were tested all around the world in very small clinical trials, usually involving 12 patients or sometimes even more. But they were very basic surgical techniques, and the methods varied greatly. But in the best scenarios where everything was logically aligned, and by that I mean the surviving transplant with dopamine cells from fetal, these are the same cells we now generate from stem cells, you could have a behavioral recovery in some patients that lasted up to 20, 25 years now. And so we were inspired by that and thought the main thing was that we could not get these cells in any reasonable amount from fetal aborted tissue. And instead, we went first through xenotransplantation. We used animal cells, and then we used embryonic stem cells. And as you mentioned, we went to iPS cells. And the personalized opportunity there is not just transplantation, as Dr. Scherer mentioned, but also we actually believe that we will be able to maybe even stem some of the underlying disease progression in individual patients if we understand their immunology, their genetics better than we do now. But we have shown up in primates now that we can develop, only if we transplant this very specific midbrain dopaminergic neurons in substantia nigra, we can get primates to recover very much like a patient would do. And so we are progressing very carefully here in Harvard Medical School hospitals to see whether we can make this in a safer and reliable protocol that the Food and Drug Administration would then regulate and help us test.
And Todd Scherer, pick up now, if you would, on the last 2 points, which is that their primary purpose would be to, if we can make this work, to generate the dopamine that goes missing in Parkinson's. Dopamine, of course, tied to the motor symptoms of the disease. And it's important to underline that, I think, for a moment, too, because we've also now learned, of course, that Parkinson's has many features that are not limited to those motor symptoms. And so that's this last point, which is that the disease is still progressing, even if these stem cell transplants in the future were able to work. So, say a little bit more, if you would, Todd Scherer, about what we hope this can do, with the appropriate cautionary note, perhaps, about what it might not be able to accomplish.
Yes, I think that the main goal of this therapy, which has been just outlined, would really be to utilize the technology of stem cells and all the capabilities of generating and targeting these cells to become a true dopamine cell, and transplant that back into the brain to replace and replenish, and in an ideal world even reconnect in the right way, as if the disease hadn't happened to the dopamine cells in the brain. So if that really was successful, you would predict that the full array of symptoms that currently respond to the dopamine treatments would be very significantly ameliorated. And while the disease would be continuing to progress, you would be starting at a new starting point where you had a much more robust population of dopamine cells that are active in the brain. So that would be sort of the ideal scenario, where you'd get an amelioration of symptoms and sort of going back in time in the disease. The caveat,
like a reset button, sort of, Todd?
I think it could be, if everything that Dr. Isaacson and others are working on is successful. The caveat, which is what you got to, is that we also now know that Parkinson's disease is impacting other brain regions, other brain systems, even systems that are outside of the brain, like the digestive system and cardiovascular system. And we're still understanding exactly what the neurological basis for those symptoms may be. But it's not clear, at least in the current setup of how we go about this treatment, that the full array of symptoms would be equally impacted because of how we're targeting the transplants, at least in the current incarnation of this therapeutic approach.
Let's move. Yeah, go ahead, Dr. Isaacson.
Yeah. So exactly what Todd said, and I think in the reasonable way of thinking about that, if you would accomplish that specific goal and accomplish it in exactly the way that we just outlined, that would be one step actually for the treatment of Parkinson's disease. What the Fox Foundation and others, including larger teams like the one that I lead, we have a very broad approach, and we are very focused on treating both this event, the patients that show up in a neurologist office, hopefully, and show these signs and symptoms of Parkinson's disease. They have lost almost exactly 65 to 70% of their dopamine neurons, and they are dead. So at that point, what we are trying to develop is to restore function. But in parallel, we work very hard on this, what we call the preclinical phase by some, pre-symptomatic by the doctors who see patients. If we could just get to finding out who was at risk, a little bit like we do for heart disease now with blood pressure and cholesterol, we would like then to treat in parallel those other systems that are at risk. So one way of looking at it is that the people who show up in a neurologist's office and get the diagnosis, they are actually potentially candidates for something that replaces dopamine, and more specifically perhaps dopamine neurons. But then all other patients, including those, are we're very eager to find ways of helping them for their specific disease. And as you may know, there is about 20 genes that predict an increased risk of Parkinson's disease, and they all relate to similar what we call biochemical pathways. protein, one particular called alpha-synuclein, but then there's 19 other genes that involve clearance of proteins and another organelle in the body that provides energy to the cell called the mitochondria. So we're very actively testing patients' cells and human cells to see whether we can discern what they may need to protect themselves against further degeneration.
Let's move to one last point, and then we want to start taking all of your questions as well. And this is really brings us to kind of where are we now, because we've been talking about a lot about what we hope this therapy can provide, but we also want to talk about, when we'll actually be able to get there. So, as we've been reviewing, we've been working on this since the 90s. Initially, as Dr. Isaacson was mentioning, they were fetal tissue, then moved on to the concept of embryonic, and now these induced pluripotent stem cells, which everyone seems to think have the most promise for actually going into stem cell derived therapy. But Tadsher, it's really important, I think, to make the point that this is long, as you were saying before, long, hard, careful science that takes time. And we're not yet at the point where clinical trials are taking place, and that's why we have this cautionary note on the screen, and I'd like you to address that if you would, Todd, because there are places that right now are advertising stem cell treatments for Parkinson's disease, and we want to make the point very clearly to people who are participating in our webinar today that buyer beware. Those are not yet. Those people who are purporting to provide that service are really not providing the kind of service or hope-for goal that we've been describing.
Yeah, so I guess just to comment on that point particularly, in my view, and I think this is shared by many, there has been no demonstrated efficacy or benefit of any of these types of therapies that are being promoted, and if anything, they may even come with some risks associated with them. So, as we discussed, there's a very rigorous process and rigorous testing that is going on to evaluate not only the effectiveness but also the safety of these treatments, and we need to be very careful to look at therapies that are shortcutting that system and making claims that haven't really been demonstrated or proven. So that I do think is, to your point, it's sort of buyer beware and really look into and evaluate anything that is being presented, particularly something that may sound too good to be true. The other comment I would make back to sort of where are we now, this is a field in terms of the particular types of transplant therapy we're talking about that I think in the last 12 to 18 months has reached sort of a new renaissance and inflection point of potential. And it's a very common pathway, unfortunately, that a lot of these research areas go through, where there's a period of really rapid advances and then roadblocks are hit and we seem to go perhaps sideways for some period of time. And in this case, we had made great progress early on in generating dopamine cells from embryonic stem cells, and then through some investigation realized that perhaps those weren't the right types of cells that we needed, and had to go back to basics. But recently, both with work that Dr. Isaacson's group has done, as well as a group in New York looking at embryonic stem cells, we are now seeing some great potential in results in some sophisticated animal studies looking at the survivability of these transplants and their ability to restore motor function in animal studies. And now we're sort of now at this new phase again of rapid progress where we're trying to convert these discoveries and move them back towards the clinic, which I think we now have some great potential to see in the next couple of years. So this is a field that I think has had challenges in the past, but now seeing some of the new scientific advances, we have the potential to be testing this potential again in the near future.
Yeah, and to
Dr.
add to
Isaacson,
that,
go ahead.
So the point that is underlined on the slide for those who are watching this webinar, extreme caution. And it mentions specifically stem cells injected in the blood to treat Parkinson's disease. You may recall one of our first slides showed you the different type of stem cells. Now, in many clinics, we have counted maybe even 15 to 20 worldwide. There is a use of what we call mesenchymal stem cells, and you can sometimes get them from blood or from fat tissue. And if there's anything that you can retain from this webinar is that what we can get from embryonic stem cells or iPS cells is a specific neuron that is now newborn. It's not going to divide again for the rest of its lifetime. That replaces that neuron that was lost in the disease. These other methods that have not actually been tested scientifically, they kind of have been in a, I have to say, my opinion or expert opinion would be that they've kind of been in a recyclable backwater for not just Parkinson's disease but ALS and a number of other diseases, where people have claimed that they can take, say, a cell from fat tissue injected in low concentration in the blood and hope to see an effect on the brain. That has not been a very active research field, but unfortunately it has come to clinics that are not under regulation by either the European agencies or the United States FDA. So many scientists, including myself, have worked very hard to try to educate the public on the differences between using stem cells. Remember, they divide, but we don't actually implant stem cells. We implant the product of the stem cell, which is the dopamine neuron. So there is a huge difference between these other stem cell therapies and the ones that Todd Scherer just mentioned, of which there are maybe 2 or 3 variants that come from embryonic stem cell-like cells. And in each one of those cases that may go to clinical trial, we have spent a lot of time trying to find out exactly how to do that right for the patient. And there's 2 very different fields, actually.
And just so that we're crystal clear about the words we're using here, Todd Scherer, we're getting a number of questions, and we're going to get to all of your questions next, about adipose stem cells. Those are those fat cells that Dr. Isaacson was just describing, right?
That's correct.
Yes. So in fat tissue,
so again, those are cells that you know, if you see something advertising injected into the blood, adipose stem cells, those are the ones that we're asking everyone to stay away from. We might as well be as blunt as that. I'm going to push us ahead to one last point and then begin folding in all of our questions, and that is, Todd Scherer, if you would please, how this research can also be used to help us learn more about the disease itself. This is the so-called disease in a dish idea. Just briefly describe what else this research can enable us to understand about Parkinson's, Todd.
Yeah, so I think there's a couple of aspects of this. One is just in the pure research that's going on around how we convert embryonic stem cells and engineer them and coax them into dopamine-producing cells, and particularly the dopamine-producing cells that are active in the substantia nigra system, which is impacted in the disease. And through that research, we've learned quite a bit about what factors are involved in promoting the survival as well as the degeneration of those cells. And that has been quite valuable to identify new opportunities to develop treatments that could increase the survivability of those cells in a disease state. I think separately and perhaps even more excitingly is this opportunity around the induced pluripotent stem cells. So we talked a lot about how you can now take this cell from a skin cell, for example, from someone with Parkinson's, and use that to make a dopamine cell and then do kind of a self-transplant back into the brain. But I think what is also incredibly exciting is that you could take that cell from someone with Parkinson's, make it into a brain cell, a dopaminergic neuron, for example, and then really try to study and compare the cell that comes from somebody with Parkinson's, which again has the DNA, for example, of the person with Parkinson's, and then compare that to someone without Parkinson's, and really try to understand then what is the impact of the disease on how that neuronal cell, that brain cell functions, and what can we do to correct whatever is dysfunctional in the person with Parkinson's versus someone without Parkinson's. Why it's so exciting and important for a disease like Parkinson's is, again, in this case, the disease is in the brain, and the brain is very hard to get a window into about what's functioning in that brain. It's not like in cancer, for example, where we could take the tumor out and look at it under a microscope and really try to understand the biochemical or cellular impacts of the cancer. We need to be creative and innovative to try to get insight into what might be happening in a pathophysiological sense in the brain of someone with Parkinson's. And this new technology around iPS cells really allows us to have great insight into what might be happening and how to correct it.
Terrific. Let's push on then and get to all of your questions. So many questions that are coming in, and really great questions. So we want to take the balance of our hour to respond to those the best we can. Here's a first one from Rob, and a number of people have asked versions of this question. We know, Dr. Isaacson, that one of the problems in Parkinson's disease, or we think we know, that one of the reasons those dopamine cells go missing in Parkinson's is that there's a sticky protein in the brain called alpha-synuclein that clumps together and kills cells. And Rob's question is, how might a stem cell approach interact with that protein aggregation?
Well, yes. So this protein alpha-synuclein shows up in patients who have either a genetic form of Parkinson's disease. So curiously, even if you have what we call a normal elevation of the protein without mutations, if you have too much of this protein by genetics, you have a very high risk of developing a very aggressive form of Parkinson's disease that affects the dopamine neurons, but also other cells in the brain that are creating risk for dementia. This particular protein is curious in that it aggregates when it gets into higher concentration inside your cell. And there are many reasons. The 20 genes I mentioned almost all converge on either not breaking it down correctly. There are various things inside the cells that clear up proteins. You can actually block those clearance mechanisms, and then you get the form of this sticky protein. What I think is perhaps not understood is that this protein actually has a normal function. It's part of your what's called the synapse. So this synuclein sticks to little vesicles that release the transmitter that we all need to function, the dopamine. And it normally works that way. With age, as we all age, say past 65 and approaching 100, hopefully, most people will have small aggregates of these proteins. Now, the question about how they interact with stem cells. Now, remember, what we create from the stem cell is a newborn neuron. Let's say it's 45 days old, which is a long, long time ago in the fetus. Now, for anyone with Parkinson's disease, even the most aggressive forms, the most early Parkinson's disease that you would find the literature occurs around 35, 40, 50 years of age, and then it's a very aggressive combined form of disease. So we think that, and we show actually that if we transplant cells into patients, even though some of those cells may age prematurely, the vast majority of them continue to function, as I mentioned. That's the evidence for up to 15, 20 years. And even if they a few percent, depending on the methods actually, in the literature it ranges between less than 1,000, 1 in 1,000 to up to 4 or 5% of the cells may form these inclusions. It's actually factually not known whether it's because the cells are not in their best position, meaning they're being transplanted to a human brain from the fetal stage. And so I actually don't see it as a major factor in this type of cell therapy, because even if you have the most aggressive form of Parkinson's disease genetically, the cells would adapt and function for at least 40, 50 years. And let's assume a patient on average would be 60, 65. That would be a fairly long time indeed. Now, I want to clarify that we know, and we discussed very thoughtfully now, that the disease progresses in other systems that may have alpha-synuclein. But I'm very hopeful that the very aggressive research that we and others do on trying to prevent those lack of protein clearance throughout the brain can be addressed in parallel to these cell therapy derived methods. So alpha-synuclein, where it is sort of the obvious culprit to most scientists, I think is really the tip of the iceberg, and we can look at all sorts of other functions that we can perhaps help the cells to survive.
Todd, just briefly go a bit further with that if you would, because a lot of people are writing in, is this a cure? And what Dr. Isaacson is suggesting is sort of what you were saying before. This could be a reset button that takes people back in time to a much earlier point, but at the same time, it may also be important to work in a parallel fashion on other therapies that, whether that's in busting up alpha-synuclein or dealing with other aspects of the disease that we know go beyond just the motor symptoms. So it's sort of like with Parkinson's, you need multiple solutions, perhaps. Would that be one way of saying it?
Yeah, I think that's right, and I think that each of these types of therapies that we've talked about would be significant and substantial steps forward, and that we would then be able to focus our efforts even more on the aspects of the disease that are not being addressed, which still would need to be addressed. So it's not that you would go to having absolutely no symptoms, but I think we talked about sort of the strengths and caveats, but just kind of at the highest level and taking a step back, I'm guessing if you told somebody who was 15 years into Parkinson's that we could put into your brain all the dopamine cells you'd kind of laid out in sort of when it might be used in the course of the disease, given other available therapies as well.
Okay.
Dave, are you there still?
Hi, this is Dave Iverson. My apologies, everyone. My everything went dead, so I had to re-enter the webinar. Apologies to all of our participants and to you, Todd, and to you, Ulla Isaacson, as well. Let's resume, if we can, with our conversation. Todd, you were describing, kind of where we are with all of this, and that we need to pursue multiple solutions to this disease. And in the interest of time, let me just move on to an additional question which relates to that in some ways. And Dr. Isaacson, let me hand this to you, which is people are very eager, not surprisingly, to participate in this kind of research. Again, we're not yet in the phase of clinical trials. Can you tell us how long it will be before you think that there will be clinical trials that will involve people being able to participate in this kind of potential therapy?
So here is how it works, and I think, in the interest of educating all of us, let's look at how the FDA, Food and Drug Administration, deals with this. And I think they actually do it very well in the United States, and Europe is very competent at this too. So what the teams out there are doing right now is submitting ideas for how they would make the clinical trials. And that would involve, in most cases, maybe 6 to 12 patients in what we call phase 1. Phase 1 is really just what the Food and Drug Administration calls a safety trial. Every agency like them who are regulating, the protection of our public against bad ideas, they will demand that the doctors that go forward with this test this very slowly and very rigorously. So typically in a situation like that, there will be one patient at a time, usually a month or so apart, to make sure that nothing surprising happens. Because this, after all, you understand, is an experiment. It's a bit like a launch of a moonshot. We don't know exactly what will happen. So before the FDA allows you to do that, or any agency actually in Europe or Japan, they will ask the doctors and scientists provide a big dossier about all the work they have done. And in fact, before you go to the FDA for the first time, you have to show usually years of research to illustrate that you really know what you're doing. And then there's a discussion with the FDA, which is called a preliminary hearing or a preliminary investigation hearing. And those are beginning to take place for a number of teams in the United States, a couple of teams in the United States, and we believe maybe one in Europe, one in Japan. So these are very important things, even though this is obviously taxing all of our patients, including the scientists and doctors, and certainly the patients. But it's really the only way to go forward, because if we don't do it this way, we end up with big risks and gaps in our knowledge. So I would say, from what I understand, and this is of course a projection and a guess, the earliest time I would imagine serious trials to be done in the United States, in Europe, would be approximately 2 years from now. And if so, those tests, I think, would be quite rapid, in the sense that you would find out within a year or so whether the treatment worked, even in a phase 1, if they were safe. And in that case, what the Food and Drug Administration and usually biotechnology industry does is to expand to larger trials. So they call them phase 2, which is now really about efficacy. How can you show that it works in many patients, and is it really worth subjecting our population and public or patients to this? And how much better would it be compared to other treatments? Because these treatments are not inexpensive at this stage, and only if we would use them in thousands of patients would they probably come down in price eventually. So I hope I've explained to you that we would project maybe 1 to 2 years, probably 2 years. And those first trials will be done with patients that were just selected from the doctors who were involved in these trials' own patient base, and randomly, frankly, to be protecting the public. And in the phase 2 screens, they would expand to other centers, maybe also in Canada, for example. But all this depends on the success of the scientists and the resources to do this work. And I'm on the same page there, I guess, as the Michael J. Fox Foundation, that research resources are scarce, and even those teams that we're thinking about usually works with more or less a limited amount of funding to get to the next stage. Definitely not ready sort of to spend all their funding on huge trials, but I would say optimistically that I would be surprised if there isn't a trial within the next 2 years. But at the same time, as hard it is to accept, it actually depends on the science that leads up to that, and a very strong interactions with what we call regulatory agencies like the Food and Drug Administration, which I actually think are doing a fantastic job based on what they know and what they try to establish.
And Todd, Chair, there are quite a few questions, and understandably at this point, this is material about which we can't have any great precise information about, but a lot of people are curious about who would be the best potential subjects for this, and would there be rule-in or rule-out criteria? For example, someone asked if you've already had deep brain stimulation, for example, would you also potentially be a candidate for a stem cell-derived therapy and for transplantation? Any thoughts, Todd, on who might be the candidates for this, and whether some people would be ruled in or ruled out?
Yeah, I think this would follow the path that Dr. Isaacson was just discussing, where I believe earlier in the process of developing the drug, the population of patients that would be targeted would be narrower. In terms of trying to really, first of all, again, as a surgical therapy, I'm guessing would be targeted more to people at that same phase where they might be considering something like deep brain stimulation. Although, just as a point, yesterday there was some news that the criteria for deep brain stimulation does seem to be moving earlier into the disease. There's also been some work to try to use neuroimaging and other tools to better select patients and the location of where we would want to place the transplants. But you're definitely going to be looking for patients that respond to dopamine treatments. Again, this is a dopamine treatment, so you'd want to find people that have good responses to dopamine treatments. And probably in the early tests, like Dr. Isaacson was saying, to look for benefit over existing treatments, that people that, while they're responding to their dopamine treatments, are having some of the side effects or the fluctuations that are demonstrating that there's a limitation of the advantages of those treatments. As the project would gain more strength and support, the population of eligible patients would eventually wind up becoming less narrow and more expansive. But given that these trials by design, because again it's a surgical trial, will be smaller than drug-based trials, the investigators will probably want to be very specific in the early testing about which patients are included. Just another point in terms of participating in research related to this topic, the research that we talked about related to the iPS cells and using iPS cells as disease models is also an area of research that people can participate in, and
right.
that really is much less involved. I mean, it's still somewhat invasive in that you would give either a blood sample or a skin sample, but that is something that also is many studies going on throughout the U.S. and in Europe trying to link clinical information from the collection of these cells and then the biological study of these cells, and that's something that people wouldn't have to wait for in terms of participating.
Again, these are the kinds of cells which, if you're someone who has Parkinson's, you could donate cells. That cell could then be teased into becoming a brain cell with Parkinson's disease and would give you a chance to really study the disease right before your eyes in a dish. And when we conclude today's webinar, we'll let everyone know how they can find out about participating in research in general, including perhaps identifying those kinds of research locations near you. A lot of questions, Dr. Isaacson, and I know you're not a surgeon, but if you could just very briefly, because we're very short on time, describe how the surgery would take place. I assume location, like anything else with this topic, is crucial. Can you just say something about what the transplant procedure would actually be like, what kind of surgery would be involved, where these cells would be placed?
Yeah, so, from growing these millions of cells in a dish, they're placed in a little test tube about, half an inch deep or less, frozen down in that liquid. And then after safety testing and definition that these dopamine cells actually are in that liquid, the patient is brought in after the PET scans that Dr. Scheer also mentioned that defines their loss of dopamine, to have them injected, and that's done under local anesthesia, very much like actually a deep brain stimulation is done. You can have a coordinate system to find small locations in the brain, and the patient is then, after a small drill hole, I have to make the analogy to a dentist because it does sound the same, but the injection is made in a very discreet way. It's actually just the amount of cells that are injected, you can probably see them as a little drop on the tip of your finger. So it's a very small amount, but obviously millions of cells can be in that microscopically. They're injected over about 1 to 2 hours into the location where the synapses usually grow. Now, if you can imagine, the fetal brain is very small compared to a grown-up person's brain. So we place the cell in close proximity to the cells where they're supposed to hook up, and the dopamine cell is a cell that creates massive amounts of synapses. And after those 2 hours of injecting a small amount of these cells, the needle is withdrawn, and the suture is closed up, just like you do for a deep brain stimulation surgery. And at that point, the procedure is basically done, and the patients would possibly have a follow-up MRI, but after that, there is a wait period of about 6 to 9 months, usually, until there is the next PET scan to establish that you get a dopamine signal from the implant. But it's a day-long procedure. Usually in clinical trials, the patient comes in the day before and has the surgery the next day, and then actually would leave on the next following day. But that's basically the procedure. I hope I explained it clearly.
Yeah, no, thank you. Yeah, that's a nice run through of what this will hopefully someday in the not too distant future be available for people to participate in. Unfortunately, we are at the end of our hour, and so I just want to get a quick last thought from you, Todd Scherer, about again this important point that keeps coming into our conversation about participating in research. And while clinical trials of this technology are not yet ongoing, we hope they soon will be. And in the meantime, there is research that people can participate in. Say one more word about that, and you see now on the screen how you can go about finding that particular research project that may be near you.
Yeah, I mean, I think just there's been a lot of different scientific hypotheses and information that were shared on this call and many of the other webinars that we've conducted. And I think it's an obvious fact, but just to reiterate it, that the only and best way that we all learn about the disease, whether it's the genetics, the symptoms, or the impact of therapeutics, is really from people who have the disease themselves and their experiences. And that is an invaluable contribution that can be made to advance research and to advance it more rapidly. And that's why we've put in place the Fox Trial Finder, which is a good mechanism to learn about trials and studies that are going on in your area. And sometimes it involves an experimental therapy, or sometimes it involves giving a blood sample or a spit sample or simple commitment. But it's something I would encourage everybody to consider, both with Parkinson's and without Parkinson's, because the only way we learn what's different about Parkinson's is also by studying and participation of people who don't have Parkinson's. And it's a major way that we can more rapidly advance our research and advance our knowledge and lead to the new therapies that we're all hoping will come more quickly than they currently are advancing.
Todd Scherer, thank you, and thank you to Dr. Ulla Isaacson at Harvard for your participation in this webinar. We did cover, I hope, a lot of important territory. We know, as always, there are questions that we left unanswered, and if you have questions that you would still like to pursue, we'd encourage you to visit the Michael J. Fox Foundation Facebook page on Monday. That's Monday, February 22nd at 12 noon Eastern, where we will take up the remaining questions online and provide the best answers we can to that. So thank you again to both our guests and to all of you for participating in today's webinar. Sorry for those audio adventures where we went radio silent for a bit, but we hope you stayed with us. We'll be sending you a link to this webinar again so that you can listen again or share it with others. We'll also have a survey link in that email so that you can share your thoughts on today's presentation. And last, we just want to remind you that we will have our next Third Thursday webinar on March the 17th, where we'll be taking up the latest techniques and research findings on how to diagnose and track Parkinson's disease earlier. Dr. Isaacson referenced earlier today, if we could get in on this earlier, it would make so much difference. We're going to talk in our next webinar about the progress we hope we're making on earlier ways of diagnosing Parkinson's disease. So join us for that Thursday, March the 17th. Thanks again to Dr. Isaacson and Dr. Scherer. Thanks to all of you for participating. I'm Dave Iverson.